Throughout this presentation, we'll be observing the possible gametes of frigate bird crosses and their probabilities of occurring using the three-point reverse mapping method. This is the genetic map of the traits of crossing a pure-breeding male with long tail, small size, and white belly to a female with a short tail, large size, and black belly. The coefficient of confidence, C, is 0.8934. The distance of R1 is 23 centimorgan, and the distance of R2 is 17 centimorgan. It's important to keep in mind that 1 centimorgan is equal to 1% recombination. In order to produce the F1 generation, or the map that we saw above, the paternal and maternal chromosomes have to be crossed. As a result, the offspring will receive each trait from both parents. If we test cross the F1 generation with all recessive genotypes, we will get 8 different types of progeny. 2 of the progeny will be non-recombinant. In other words, they will be the exact same copy of their parental generation. The rest will be recombinants. There are 4 types of recombinants that we observe. The first is the double crossover. The second is the single crossover 1. The third is the single crossover 2. Lastly, the fourth is the no crossover. And note that these are the exact same copy of the parental generation. We can also calculate the probabilities of each progeny occurring. We multiply each progeny by 1/2 to account for both alleles and get the probability for each recombinant type. In order to calculate the double crossover, we calculate the coefficient of confidence first. Then we multiply R1 times R2 times the coefficient of confidence to find the double crossover. In order to calculate the single crossover 1, we subtract R1 minus the double crossover to cancel out everything else except R1. This process is the same for the remaining 2 types.